orchards. The obscure mealybug is a polyphagous agricultural insect pest that attacks multiple fruit crops, and in South Africa is one of the dominant pests found on both plum and stone fruits. When it feeds, the mealybug, it excretes a sugary substance known as honeydew. This honeydew acts as a substrate for sooty molds, and then this mold contaminates the produced fruit, and the presence of the mold on the fruit affects its marketability. And also sometimes the mealybug infests the produced fruit, and the presence of the mealybug within the fruit or in fruit consignment affect their export because it has serious phytosanitary implications. So the aim of my study was to evaluate the potential of South African entomopathogenic fungal isolates as potential biological control agents of this mealybug. We did this by screening local EPF isolates against this mealybug in the lab, and we selected the most effective isolates, and then we mass-produced them, and then we formulated the mass-produced conidia, and then further tested the formulation against the mealybug. For our screening bioassays, it showed that 2 local Metarhizium isolates, mainly Metarhizium pinghaintii and ruberti, were the most effective isolates against the mealybug, as they both induced average percentage mortality of above 90%. The pictures that you see here are the infected cadavers of the mealybug following exposure to the EPF isolates. And then the diagram that you see here is the mass production protocol that we developed in the lab to try and mass produce the conidia of the 2 most effective EPF isolates. We mainly used the blastospores of the EPF isolates, and we used rice as an inoculation substrate to mass produce the conidia. We further formulated the mass-produced conidia using various oils as carriers and also diatomaceous earth. And when we assessed the results, it showed that to formulate the conidia of the mass-produced isolates, the oils were showing they were more effective in maintaining the pathogenicity and the virulence of the pathogenicity and the longevity of the conidia relative to storage of the conidia using diatomaceous earth or storing the conidia as it is. And when we tested the formulation after a certain period of time, we waited 2 months and tested the efficacy of the conidia that has been formulated in oils, and the conidia showed to induce mortality in colonies of the mealybug following storage after a long time. So our study showed that local EPF isolates, mainly the Metarhizium pinghaintii and ruberti, can be used as potential biological control agents of the obscure mealybug, and that those isolates can be easily mass cultured using rice and blastospores as inoculum. And also the mass-produced conidia can be formulated in oils to preserve them for long-term storage. And therefore, our study has contributed to the potential biological control agents that can be used or further explored for control of this mealybug in deciduous fruit orchards. Thank you.